Every day, billions of cells divide in our body to grow, repair tissues, and replace old or damaged cells. This biological process, known as cell division, is the basis of the reproduction of living organisms. An extraordinary, precise, and almost perfect mechanism. But what happens when something goes wrong? When a small mistake, seemingly insignificant, upsets the balance? It is precisely at this moment that a tumor cell can be born, the first spark, responsible for the onset of a complex and dangerous disease, cancer. Our body is made up of approximately 37 trillion cells, within which we can identify the cell membrane, which isolates it from the external environment, the cytoplasm, where most cellular activities take place, and finally the nucleus, the cell's command center. It is here that we find DNA, a sort of instruction booklet necessary for the cell to be born, survive, and correctly carry out its functions. Under normal conditions, through a process known as mitosis, a stem cell divides to form two daughter cells, genetically identical to the progenitor. Their nucleus, therefore, has the same chromosomal set as the stem cell, that is 23 pairs of chromosomes, within which the genes are contained. This process, known as replication, or DNA duplication, is one of the most delicate phases of cell division, as errors may occur that give rise to mutations. In this case, we speak of spontaneous mutations, and they occur following errors made randomly by cellular processes within our organism. In the case of induced mutations, however, the modification of the DNA is due to the action of a factor external to the cell, called a mutagenic agent, for example cigarette smoke, alcohol, X-rays, and ultraviolet rays. In most cases, the healthy cell is able to recognize and correct this continuous and daily damage thanks to a series of DNA repair mechanisms in which up to 100 different proteins intervene. These defense systems have in fact been defined as the first aid kit of our genome, active 24 hours a day, ready to repair damage to our DNA. However, if the cellular damage is too extensive or the errors are difficult to repair, apoptosis occurs, a natural enzymatic process, genetically controlled, which leads to the programmed death of the diseased cell. In some cases, however, the repair mechanisms do not work as they should and cause the mutation not to be corrected, but to remain in the cell's DNA. At this point, the information contained in the DNA is incorrect, so the cell can start producing unnecessary proteins, synthesize them at the wrong time or in the wrong place, or synthesize them with a different structure than expected. Although not all mutations are harmful, each of them represents a potential danger for the individual, especially when they affect those genes involved in the control of cell growth and apoptosis. Under normal conditions, proto-oncogenes are genes that regulate cell growth and division in a controlled manner. However, when they undergo a mutation, they transform into oncogenes, pushing the cell to divide in an uncontrolled manner, thus activating those processes of cancer development. If oncogenes are the accelerator of the cell, oncosuppressors can be compared to brakes. These are those genes that are generally involved in the repair of errors in DNA and in the processes of apoptosis. However, when they mutate, they can be inactivated or even eliminated, thus allowing the proliferation of the tumor mass. At this point, the cell with the damaged DNA, if able to divide, will pass the mutation onto its daughter cells, which in turn will pass it on to subsequent cells. Over time, an ever-increasing number of mutations will accumulate, which if they affect genes involved in the control of cell division, programmed death or DNA repair, can cause the onset of cancer. This process, called carcinogenesis, transforms normal cells into cancer cells, leading to rapid uncontrolled cell proliferation. The tumor mass will then begin to grow, but to do so, it needs oxygen and nutrients, so it will begin to produce some substances capable of stimulating the formation of new blood vessels. This process, known as angiogenesis, allows the creation of a network of vessels, starting from pre-existing ones, playing a key role in the development and growth of the tumor. Furthermore, tumor cells can also break away from their site of origin and migrate through the bloodstream or blood vessels to other parts of the body where they can form a new secondary tumor. This process is called metastasis and is one of the distinctive characteristics of malignant tumors, responsible for approximately 90% of cancer deaths. In conclusion, we have seen that cancer is not a sudden event, but the result of a slow and long process that originates from alterations in DNA. However, it is believed that a single mutation is not sufficient to trigger carcinogenesis, but that multiple mutations are needed to induce neoplastic transformation. These damages therefore tend to accumulate silently for years before the tumor becomes clinically evident.